Hello, welcome back to studying microbiology book. So, how are you guys? And did you already studied this book before me or alright, we are starting with the energy. We already went through Koch postulates. Wait. Okay, I was just checking. We are going to close this one and we are going to go to our PDF. So we already went through the Koch postulate and the molecular postulates of the Koch and how he developed techniques for culturing microorganisms. We already went through pasteurization, we went through spontaneous generation, we went through several scientists that did a lot of basic things in the microbiology. Now we are going towards the advancement in the golden age of microbiology, and I think they are starting with the immunological studies, since they are related to the diseases that were already being discovered by the microorganisms. And for now we were discussing things about bacteria and fungi. I think moving further from that, we are going to have the viruses. So viral pathogens were also studied during this time. The discovery of viruses. So we are starting from here, right? We already studied that Richard Petri discovered the Petri dish. Now viral pathogens were also studied during this time. The discovery of viruses and their role. Wait, let me move it further so we can see it. Was made possible when the Charles Chamberlain, one of the Pasteur's associates, constructed a porcelain bacterial filter in 1884. Bacterial filter, okay, this just means that this filter do not allow the bacteria, like it is specifically for bacteria size. So the Dmitry Ivanovsky and the Martinez Bajornik. Okay, do you have any familiarity with these names? We already studied them in the landmarks of the history. They used the filter to study the tobacco mosaic disease. Like, the advancement in the bacterial diseases were progressing, so that thought might be that the tobacco mosaic disease was also because of the fungi or the bacteria. But when they used the filter, they found out that the plant extract and sap from the diseased plant were infectious even after being filtered with the Chamberlain filter. How do they know that they were infectious? Like, this is the common knowledge in the microbiology or the biology history or the viruses, that the infectious agent passed through the filter that was designed to trap the bacterial cells. The agent must be something smaller than a bacterium. Bajornik proposed that the agent was a filterable virus. Eventually viruses were shown to be tiny acellular infectious agents. So in this paragraph we just studied that there were two scientists, right? First was a scientist, the Charles Chamberlain. He constructed a porcelain bacteria filter in 1884, while the Dmitry Ivanovsky and the Martinez Bajornik found that there are organisms that are smaller than the bacteria that can cause diseases. I'm going back because I want to see where was the name of the Bajornik in this. Here there was some place here in 1899. So I want to just compare it to the age where the associate discovered or made the porcelain bacterial filter. Wait, here. Alright, so this is 1884 and that was 1894 or something. So like after 10 years, maybe approximately 10 or maybe more than that. So then what happened? Immunological studies. In this period, progress also was made in the determining how the animal resisted disease and in developing techniques for protecting humans and livestock against the pathogens. So like in this era where the viruses were discovering, the diseases were discovering, postulates were coming, people were also developing determining that how animals resisted disease. Like they were studying the resistant mechanism. And you know that our immunology, no, sorry, not immunology, our immune system act as a resistance system to the foreign particles, and they were developing techniques for protecting humans and livestock against pathogens. Like they were developing techniques to develop that resistance, or to learn how to develop that resistance. During studies on the chicken cholera, again the Pasteur, I thought we were over with the Pasteur. Pasteur and Roux discovered that incubating their cultures for long intervals between transfers would attenuate the bacteria, which means they had lost their ability to cause disease. Alright, so they are saying that incubating them for long intervals between transfers will make them lose their ability to cause. Maybe there will be something change in their physiology here. If the chicken were injected with these attenuated cultures, they remain healthy but develop the ability to resist disease. Yeah, even now, if they were infected with the bacteria that had the ability to cause disease, and they were attenuated, right? Still, the chicken would show resistance to them because he already learned the mechanism of the previous bacteria that was similar but lost its ability to cause disease. Then he called the attenuated culture a vaccine. Okay, he just named it Weka cow. What cow? What was it again? Cholera. Livestock, chicken cholera. It was chicken cholera. So why in honor of Edward Jenner? Because many years earlier, Jenner has used material from the cowpox lesions to protect people against smallpox. All right, all right, all right. Like it wasn't the Pasteur that was the first one to demonstrate or to learn that if we inject the microorganisms that were causing diseases, and we just make them lose their ability to cause disease and inject them into the humans, they won't cause disease. It was Edward Jenner first that did it with the cowpox viruses, and he would take the viruses from the cowpox lesions and inject them into humans in a small quantity so they could develop a resistance against them. Shortly after this, Pasteur and Chamberlain developed an attenuated anthrax vaccine in two ways. We already know that Pasteur developed a vaccine, but the Chamberlain was with it. By treating the cultures with the potassium bichromate and by incubating the bacteria at 42 to 43 degrees centigrade. Guys, 42 to 43 isn't your normal temperature for bacteria. Like the optimum temperature for bacteria, usually pathogenic bacteria usually range within human temperatures like 37 temperature. 42 to 43 is quite high for them, and their enzymes don't work. So first they treated the culture with potassium bicarbonate. Potassium bicarbonate made the cell, non-functional. Wait, what does the potassium bicarbonate do? Let me search it a little bit. Potassium bicarbonate do what to a cell? Helps by shifting the potassium into them and regulating pH, transport other ions and organic substance. Wait. I wasn't talking about their positive. In a negative way. What are the negative effects of a confusion faster to bacteria? Why aren't they understanding this? Inhibits bacterial growth. Increase the bacterial susceptibility to killing through increasing. Alright, so they increase the bacterial susceptibility, means they sensitize them to the killing. Not sensitize, weaken them so they can be killed easily. How do they do that? By increasing the bacterial CAMPs, cyclic adenosine monophosphate levels and disrupting the pH gradient of the proton motive force across the cytoplasmic membrane of gram-negative and alright, so they just change the CAMPs and disrupt the pH gradient. I forgot what the CAMPs were doing. Do what? Is a second messenger that performs many functions in the body. It's involved in regulating. We are not talking about the function. Like CAMP actually do something, activate protein kinases and channels like HCN. Alright, it binds to the channels so to regulate them, and they activate protein kinases. Kinases put the phosphate to the material, like in this case it is protein, and the inhibitory synaptic plasticity of the photosynaptic GABA receptor. What the heck is this? We are not diving deep into this. Its time will come. Anyway, we just know that the what are we talking about? Yeah, potassium bicarbonate. It make the bacteria susceptible to killing. And they are saying that in this 36th chapter we will be studying about vaccines and immunization. So Pasteur next prepared, I think the history should be studied, in the last of the book, since we don't know the terminologies and we are already discussing it. So Pasteur next prepared rabies. All the work was done by Pasteur till now. Next, prepare rabies vaccine by different approaches. The pathogen was attenuated by growing it in an abnormal host. Okay, so there are ways to make them attenuated, like for example, changing their temperatures, putting them in a potassium carbonate, and putting them in an abnormal host. The rabbit. I think rabbit isn't a organism, a host for rabies. After infected rabbits had died, their brains and spinal cords were removed and dried. During the course of these studies, Joseph Meester, a 9-year-old boy who had been bitten by a rabid dog, was brought to Pasteur. Since the boy's death was certain in the absence of treatment, Pasteur agreed to try vaccination. Alright, since the boy was going to die anyway, so Pasteur said, why not experiment it on? There may be a chance for him to live. Pasteur agreed to try vaccination. Joseph was injected 13 times over the next 10 days with the increasingly violent preparation of the attenuated virus. He survived. Well, they didn't know the mechanism, but they said, why not we try it? In a gratitude for the Pasteur's development of vaccines, people from around the world contributed to the construction of what? Pasteur Institute in Paris, France. One of the initial tasks of the institute was vaccine production. Oh, I didn't know there was an institute named after Pasteur. Pasteur Institute in France. It looks like let me enlarge it. So is this the Pasteur Institute? It looks like it's been constructed over the previous one. This doesn't look like it's from the old age. Well, they weren't that old. Who is the who is the head of? Let's see who is it now. Professor Yasmine Belkhair is the head of Institut Pasteur in France. She became why did she become head? Non-scientist who studies the relationship between microbes and immune system. She earned her PhD in immunology from the Institut Pasteur. Oh, so we can study PhD in the Institut Pasteur. Okay, we understand it. Who was her again? Yasmin Belkaid. Okay, let's go back to studying our original work. So, after the discovery that the diphtheria bacillus produced a toxin, Emil von Behring and the Shibasaburo Kitasato injected the inactivated toxin in the rabbits. Diphtheria bacillus produced toxin. Okay, so now they found out that the bacteria could cause death due to toxins as well. So the Behring and the Kitasato injected the inactivated toxins into the rabbits. Inactivated toxins. I don't know how they deactivated the toxins, inducing them to produce antitoxin, a substance in the blood that would inactivate the toxin and protect against the disease. Like they inactivated the toxins maybe by heating it, or toxin may be in a protein structure or organic structure that could be disturbed by the heating, or maybe some chemicals inactivated it. So they put it into the rabbits so they could get the antitoxins. A tetanus antitoxin was prepared, and both antitoxins were used in the treatment of people. Okay, so the Behring and the Kitasato did produce antitoxins for which and which bacteria? Diphtheria bacillus, right? And a tetanus. Alright. The antitoxin work provided evidence that immunity could result from the soluble substances in the blood, now known to be antibodies, humoral immunity. So now antitoxin work. Like before that, they didn't know that the blood had some kind of, like cells are one thing, right? The white blood cells and these kind of cells are one thing that could, give immunity to them, like producing by vaccination, right? But they didn't know that blood had something more than these cells that could give immunity. And this provided the evidence that immunity could result from soluble substances in the blood, now known as antibodies. At first, it was just a heat labile body. And humoral, we call it humoral because it moves around the body, like antibodies don't stay in one place. It becomes clear that the blood cells were also important in immunity, cellular immunity, when Elie Mechnikov discovered that some bloody leukocytes could engulf disease-causing bacteria. What? I thought they would know that before. Okay, maybe they didn't know, because this sentence is showing that they didn't know that there were blood cells that could be important to immunity. So, okay, it's after we found out that there were antibodies in the blood. Well, of course, it was an antibody, named antibodies at that time. And after that, due to Elie Mechnikov, we found out that there were some blood cells that could cause immunity as well. And how did he discover that there were some blood leukocytes that could engulf disease-causing bacteria, known as phagocytosis? Well, here it is written. He called these cells phagocytes, and the process phagocytosis. Greek, phagen, eating. Okay, phagen means eating. So phago means eating, cytes means cells. So eating cells, you can say it, cells that eat. Okay, now what? So this was all about immunology. They said there wasn't much about the immunology. They were just saying that why is it the androgena? Yeah, Pasteur. So this was all the work by Pasteur, and in the end there was Behring and Kitasato for antitoxins and Elie Mechnikov for the blood leukocytes, and yeah, humoral antibodies as well. Alright, let's repeat it just once in a minute. The Pasteur and Roux discovered the vaccine for what? Rabies, and the not rabies. The first one was the chicken pox. Do you call it chicken pox? Chicken cholera. Sorry for me forgetting about it. So the first one was chicken cholera. They developed a vaccine, and they named it vaccine because of the Edward Jenner that was working with the cow lesions, and not antibodies, the microorganisms. And then he produced the rabies vaccine because of a 9-year-old boy. We like knew how it worked. And then the Institute of Paris, sorry, Pasteur Institute in Paris was made, and then Kitasato and Behring produced antitoxins against the bacillus, diphtheria bacillus, and the tetanus. Then the antitoxin showed that we had humoral antibodies in our body that could produce that could give us immunity, while the Eli Mechnikov also discovered that some blood leukocytes can give us immunity as well by phagocytosing. Right. Now development, and should I do it in a in a separate video? I think I should. Oh, wait. I think I should do it. Alright, I'll end this one and I will be making another one for the further topic.